it like a million times, but never got to ask you out. What if you don't feel like I do? Would I stand it like a fool? I've never met a girl like you. You turn my sky from gray to blue.
Okay, so let's talk briefly about how this so-called differential gene expression is accomplished. So what is differential gene expression? It is the core mechanism enabling the differentiation of multicellular organisms. Like differentiation means starting from a single cell, the fertilized single cell we call the zygote, and through cell division you have multitude of many cells. So these are all multiple different. So for example, how many cells do we have as a human? The average human, several trillions of different cells. So it's a huge number of cells, but all these cells are direct copies of a single cell, the one cell. So in theory, all these descendant cells in multicellular organism each different cell is directed descendant of the original one single cell, which means all these cells inherit exactly same type of genes. So these are actually all the clones. So they should be identical if they have exactly the same genes in same status as the starting original cell, then every cell, by virtue of having exactly the same genes, should be identical in appearance, how they look, and also in their functions. But in reality, things are not. So all these cells in eukaryotic multicellular organism, they differ. So that's why they have this unique feature of tissues. Different tissues. What do you mean by different tissues? Different types of tissues look different and have different functions. And further, different type of tissues get together to organize some structure, organization unit called organ. So these are the essence of the so-called differentiation. Then what is behind this differentiation is this. The differential gene expression is the key to the differentiation. So here, differential gene expression means different cells have different sets of active genes. In other words, not all cells have I should just back up. Not all genes are active in every different cell. Some genes are only active in very specific type of tissues, whereas some other genes are active in the other type of cell, and that's what makes these differences in terms of different tissues, and go further, different organs, like that. So then how do you make? You have the same gene. So let's go back to the same old classical example of this hemoglobin. Why? Even though you have hemoglobin genes in everywhere in your body, but that gene is only making the RNA copy, the transcription, only in the red blood cells. Actually, to tell you the truth, that the gene making activity, the activity of the hemoglobin gene, goes only so far as like when at a certain period of this time, then they stop, okay, being active. So this activity of the gene also can be demonstrated over different time frame. So not only this, the location wise differences in gene activity, but also the timing wise gene activity differences can also be found. So these are all they assimilated in this example of so-called differential gene expression. The key here is what the factors that make this differential gene expression possible is transcription factors. They are the one needed for the gene activity. Without them, the gene cannot be active. So here the secret is by adjusting the availability as a hemoglobin gene. Okay, if I want to become active, because RNA polymerase is everywhere, and every gene, if we define the gene as any area of the DNA that can, is not M, or is so, as a gene, as long as you have a potential to make the RNA copy, then you are a legitimate gene. However, the potential doesn't mean that you are actually active at this point. So in order to become an active gene, then what you need is, in addition to the RNA polymerase, the main catalytic enzyme for this business, but you also need several, or at least one single kind of a specific transcription factor. Mind you, different genes require different types of transcription factors, although some transcription factors can be shared by a number of different genes though. Then the way you make certain genes active and some other genes are inactive according to along with some other different location of your body is you make this needed transcription factor available in a specific tissue, and then that gene will be active in the tissue. Whereas if you get rid of this availability, there are several ways of you make this availability issue accomplished, then that gene in that particular tissue will not be active. Is the key of this this activity of a so-called differential gene expression. So this differential gene expression can be demonstrated in the case of a so-called tissue-specific gene expression, which means different tissue have a different set of active genes. Or it can be also further demonstrated in a developmental specific gene expression. In other words, this is the timing issue. So certain genes are active only in a certain timing of your development. Like at the early stage, when you're a baby or an early infant, that certain set of genes are highly active. But then as you grow older, those genes are shut off. And on the other hand, some other sets of genes may actually start becoming active. So this type of timing wise differences can also be possible. And here let me show you an example of this tissue specific differences in this gene activity in this example, like taking these two example of two protein gene. One is the albumin and the other one is the crystalline. These two genes are everywhere in our body. The albumin is a protein which is abundant in our blood, but that albumin protein is actually made in the liver and then transported out of the liver, and then they patrols, so to speak, patrols our bloodstream, and it has a very specific functions there. So one of the most abundant proteins in our blood. But the point is, it is made only specifically in the liver. Even though we have these albumin genes in our muscles and on other types of body cells, but the gene there is not active. On the other hand, the crystalline protein is the specific protein needed for our lens function in the eyes. So this crystalline gene is only active, even though that gene is also present everywhere in our body. It is only active in the specific type of cells, the crystal, the lens cells in our eyes. Then what's the difference? So this is the case. If you take a look at further, this is a kind of a summarized figure showing the difference between these two genes structure. So both the coding region, these are the coding region, of each gene, and you do have a same type of promoter, right, marked in same color, like light green. But in other than this promoter area, other control elements in the upstream of these coding regions look a little bit different. So this is the case that showcase. So you have enhancers, but with a different color coding means transcription factors. Remember, enhancers are sequence element which is very specific to each different type of transcription factor protein. That means the albumin genes require the transcription factors that are required for the albumin gene activations are different from those of that required for the crystalline gene activations, right? So two different kinds of sets of the transcription factors you have, but then what you control over here is, so like in this figure, take a look at this liver cell. What's going on? So by now you hopefully understand, in the liver cell both the albumin and crystalline genes are present, but the difference is the availability of the transcription factors described as activators over here. These are the transcription factors. That specific proteins. And here in this figure, the small grape shaped the balls are the ones representing the number of different transcription factors that are initially associated with such different specific enhancer element present in the control region of this albumin gene in the liver cell. And this is the RNA polymerase. And this is exactly what's going on. Like after binding to specific enhancer element, these transcription factor proteins collectively stabilize the RNA polymerase so that this gene expression can be efficiently processed. On the other hand, what's going on in the area of this crystalline gene in the liver cell? Here is empty. This enhancer, these enhancer DNA elements are for recruiting the different types of transcription factors that are needed for the gene activation. But you don't have this. No transcription factors are available here in the liver cells for this binding. Then if you don't have any transcription factors, then no RNA polymerase can stably bind in the promoter region and then start the transcription. So that's why no gene, so this crystalline gene is not going to be expressed whatsoever. And that is the reason why this albumin gene is the albumin protein is made in the liver cell, but the crystalline protein is not made in the liver cell. And by contrast, in the lens cell of your eyes, the opposite scenario, opposite situation is in play in the lens cell, so that here you don't have any. This area is empty, which means even though you do have exactly the same sets of DNA sequence, the enhancer element sequence is present as is the case in the liver cell, but no available transcription factors corresponding to this enhancer element. Somehow, there is a trick. It's a kind of a difficult thing to explain further, so I will just simply not going to touch. But the thing is, yeah, those transcription factors that were present in the liver cells are not available in the lens cell. So as a result, no RNA polymerase can stably sit on this the promoter area to start the transcription. So that's why this albumin gene is not going to be expressed to make any albumin proteins. On the other hand, these transcription factors, although here for simplicity, the type of transcription factors that are binding to this crystalline gene promoter area look exactly the same as the albumin gene, the regulatory element area, but in reality, so those transcription factor proteins are basically different types than those acting on the albumin gene control. So anyway, so the point is here, these transcription factors are available. So even though in the liver cell you do have these specific enhancer elements that acting as a binding site for these corresponding transcription factors, but there no transcription factors were available, so nothing happens. But here, fortunately, in this lens cell, these particular transcription factors are there available, so that they bind to this specific enhancer element. Then what happens? If you do have, then as you have seen in this case, these transcription factors can stabilize the RNA polymerase. So that's how this crystalline gene can be expressed to make the protein. So that's the basic, very simple explanation of how this differential gene expression on the tissue-specific level, tissue-specific gene expressions example. If you expand this example in the case of developmental specific expression, you should be able to explain taking this same type of example. The only difference now is like you apply this example over the different time scale, like in the same place. You don't have to make these two different locations example, but in the same type of a cell. But at the early stages of your development, you make certain gene, the transcription factors available, while the others you do not make them available, so that you can adjust this. Only specific sets of genes are being active. Then afterward, later on your development, the later time, then you just reverse this scenario. Then the other different set of genes are now active, and the former, the ones active genes are now shut off. So it's not really difficult to expand this example in the developmental specific gene expressions case. Okay, so that's once again, although it may not be such a very easy concept to understand, and in reality, the scientists themselves have only very short history of finally grasp this concept of making this available, different availability of the transcription factors, and that's the main so-called this differentiation is achieved. And actually, the further, all this thing is closely connected with the concept of epigenetics, which is epigenetics is in reality is nothing but how like adjusting this the chromatin condensation. And remember, these transcription factors can make the different degree of chromatin condensation. So certain transcription factors make the heterochromatin area changed into euchromatin area and vice versa. So combined with these two functions, the certain transcription factors make this key roles in this type of gene regulation in control. What type of genes are going to be selected to be active, and whereas what types of other genes are going to be silenced is how things are going. Okay. Now let's take a look at this splicing of eukaryotic messenger RNA, as briefly mentioned in the previous session. The initial or the primary messenger RNA of eukaryote further processed in the nucleus to finally have this shorter product. Why does it become short product? Because of this removal of introns, and the remaining parts, the exons spliced together. This is essentially what's happening for this RNA splicing. So altogether, it's called the splicing of eukaryotic messenger RNA. So this most eukaryotic messenger RNA genes are like interrupted with the introns. Introns are there, but they are going to be removed finally. So this process is called RNA splicing. And this RNA processing is one of the three major events of collectively called eukaryotic messenger RNA processing, although the other tools are not going to be mentioned here in this classroom. So basically what's going on is, if this was the primary so-called pre-messenger RNA, primary RNA, which is unprocessed, and three different types of processing have to be applied before this final version of the messenger RNA. This is a mature final messenger RNA that are present in the cytoplasm. Which is ready for the next step of this gene expression, the final version, that converting this information into the protein products. So now all that remained in here is all these introns are all gone. So the exons are the ones only have left as a result of this RNA splicing. And there is additional some part is attached on the 5' end, which is called the 5' cap. This is a kind of unique chemical structure. And also at the tail, which is a 3' end of this RNA, some, so to speak, artificial, no, it's not human made, so I shouldn't say it's an artificial, but additional mysterious the sequence consisting only of a adenine residue of RNA, so we call it a poly A, like A, like you can also like something like 20 or 30 to several hundred, like 200 repeats of A sequences is attached afterwards. So these two are called 5' cap and the poly A tail respectively. So these are 1, 2, 3 of the major RNA processing of eukaryotic messenger RNA. And why there has to be a 5' cap and 3' and the poly A tails, scientists kind of pretty much understand and the reason and the function of this. But the case of this introns, why there is an introns? Well, like in retrospect, so like there are two schools of thoughts, sharply divided and like debating still the on the origin of the introns. Because why do we talk about this origin of introns? Because seemingly on the surface, the presence of introns appears to be kind of a waste. So in terms of throughout the course of evolution, every different individual organism tries to be more effective, efficient, to increase their like competitiveness in this under the environment, is the fierce, harsh environment of governed by natural selection. So if this seems to be a kind of a meaningless nonsense, waste, and why do we keep these introns? In fact, actually, these introns appears to be the thing of a way long past in the case of prokaryotes, the prokaryotes bacteria, in particular, the bacterial cells don't have any such a thing like introns. It's not because of they are like relatively the less advanced creatures in terms of their evolution compared with the introns, but they have decided to become more efficient. So what's the nonsense of this having this introns? So we are going to get rid of everything. So, and it appears that that is the reason why these bacteria don't have any introns in these days. The proof, the archaea, another type of prokaryotes, archaebacteria still have introns. So maybe it's just the heritage of our lineage of evolutions. If we accept the fact that all these eukaryotic cells have derived from a certain lineage of archaebacterial prokaryotic cells. So that's the reason, maybe, why these, even though they are also prokaryotes, but archaebacteria did not entirely dump this meaningless, apparently meaningless waste garbage of introns. But anyway, the origins, regardless of which one is true in terms of this, the explanation of the origin of introns, but neither the group can clearly explain the reason why we still keep this intron. But one clue is the utility of having this intron in eukaryotic cells is this. For example, wow, having this intron, and then later on, by playing this removal of introns differentially, what we can achieve is we can take advantage of this intron generation in the event of a splicing. We can create from one gene several different kind of products in terms of final messenger RNA by adjusting, like making changes in which ones are going to be introns and which ones are going to be exons, for example. Then the sequence of final messenger RNA would be different among these three different versions, A and B and C, and each of which will generate slightly different version of protein. So more than one version of proteins out of a single gene is the thing that seems to be clearly a kind of an advantage that these eukaryotic cells can enjoy over those bacterial cells which have only single gene produce one protein. Well, actually, that is not exactly entirely true, but I will stop talking more about, because it will make things further complicated. So I will shut up at this point. By splicing different segments of exons under different conditions. For example, this is a real life example happening. One primary transcript is over here. Here in numbered block, number 1, 2, 3, 4, 5, 6, 7, 8, 9 all represent different exons, and this light gray block area or introns. So these are introns and these are the exons. However, for example, in this particular example of this gene, the exon number 6, depending on the different condition, this exon number 6 can be included in the final product or not. And the consequence is that will make the difference in whether this protein product can go into the cell membrane, or this protein product will remain in the cytoplasm. And as a consequence, when these proteins are present in the cell membrane, like this membrane protein, and as a consequence, the event is a cell death, so-called programmed cell death. So this cell will die. But when this protein remains in the cytoplasm, this cell is not going to be subjected to the death. So depending on what type of splicing event the particular cell goes through, will determine the different fate of this cell. So for example, in this example, the exon number 6 is included, and due to that, this protein will become the membrane protein, whereas when during the splicing event, this exon number 6 now, in this occasion, has become an intron, so which is not going to be in the final product, then this protein does not go into the cell membrane. So as a consequence, the result will inhibit the cell death. So this type of alternative choice of which specific block of the gene to become an intron or an exon is called the alternative splicing, right? So these eukaryote cells, including animal, plants, and fungi, all they exercise the highly expanded or broad spectrum of this alternative splicing. So you can find everywhere this practice of alternative splicing. One very showcase as an proof of example is the human genome. When human genome, before the Human Genome Project, which is the effort to decipher what kind of genes or whole sequences, the order, as I have mentioned earlier, the whole essence, core of the activity of Human Genome Project is to read all the DNA sequence of more than 3 billion nucleotide pairs of entire human genome. So then afterwards, we can identify at least it's a fairly good idea about which area represents genes and which areas represent non-gene, so-called this junk area. Before the onset of the Human Genome Project, which took 10 years, scientists believe that the number of genes present in a human cell should be at least 100,000. How do you know? Because before the actual DNA sequence investigation, scientists have already identified about 100,000 different proteins. So if one gene is responsible for one coding of protein, on top of that, actually, there should be some other additional non-coding genes like ribosomal and all kinds of different transfer RNA genes altogether. So at least it should be more than 100,000 different genes. But then after completion of the Human Genome Project, what it turns out that is, wow, we have only about 222,000 protein, which is way below the conceptual minimum number of genes. What a surprise. then there appears to be, okay, so what is going on? How do you make this thing happen possible? So this is an example of alternative splicing. So in other words, in order to make this whole discrepancy possible, it's not something one very peculiar, rare occasion of an example of one gene can make a different version of proteins, but this practice of alternative processing is widespread all over the different types of genes. Then only it is possible to make with only 2000 gene to encode way over 100,000 different proteins instead. So at least, even though we don't know the reason why we still keep this introns, but after all, the presence of introns are not entirely a total waste, because we can make use of this. But it does not say that that is the reason why the introns are still present. One cannot actually have this draw such conclusion out of this observation. But at least we can take advantage of this in this way. Okay, so next session, here we are going to take a look at the actual process of now the conversion of the RNA contained, the information, genetic information contained in the RNA into the protein building process. So this overall, that will conclude so-called the concept of central dogma, which was formulated by the Francis Crick, the other guy of the DNA, those with the James Watson and Francis Crick are the two scientists who elucidated the DNA structure, and then as a consequence they won a Nobel Prize, as I went over earlier in this chapter. This is nothing but just explaining the way of direction of what information flow in protein synthesis. You have to start from DNA, and then that information first has to be handed over to RNA, and then the RNA from RNA to protein is the way the information flows. More importantly, the hidden message, the hidden significance of this so-called central dogma is you cannot go reverse. So you cannot start the protein to make this RNA, and this RNA to make this DNA. So there is no way you go this reverse direction because you are not a salmon. So that's the point of this central dogma. But in real, actually it turns out that some organisms, notably viruses, certain viruses can laugh at us. So certain type of viruses which has this RNA genome, they can make this process, RNA to DNA. So this type of viruses are called the retrovirus. That includes the famous HIV virus, which is responsible for the symptoms like the AIDS. And these kind of viruses, thanks to the presence of some specific enzyme, its name is reverse transcriptase. Once they are in, after infecting the human cells, once they are in, these RNA virus RNA is a single strand, the genome. And these RNA viruses, their strategy to survive as a parasite is convert their genome in temporarily into double-stranded DNA and then insert their genome into some any random location of a human genome. So like human cell or any other animal cell. Yes, RNA, the retrovirus, this RNA genome, it converts into double-strand DNA form. Okay, and so this is nucleus, and we do have our own genome, DNA genome. Okay, yeah. Let it insert their part of gene over here. So this is their lifestyle, and for that, obviously, the specific enzyme, reverse transcriptase. Ah. They exist. And actually, the reason why I specifically mentioned this enzyme is this has been borrowed by the scientists in genetic engineering. And has made a huge contribution in the advancement of the so-called genetic engineering or the recombinant DNA technology, like together with the restriction enzyme that I mentioned earlier in this chapter. So the restriction enzymes were borrowed. Borrowed is a nice word. We stole without any permission, just like Watson and Crick. So this also, this particular enzyme was also stolen from this retrovirus. That has been a very good utility tool in all kind of different activities in progress of genetic engineering. But then actually it turns out that earlier it was a thought. Dinner is ready, my wife's. The scientists believe that, okay, so this is the kind of a unique feature only present in this particular type of a virus, like a retrovirus. But then it turns out that like humans and any other advanced eukaryotic organism to actually have their own kind of version of reverse transcriptase. Yeah. Maybe if we can have an opportunity of mentioning this, our own version of reverse transcriptase or not, in maybe following chapter when we have a chance to talk about the telomere. One of the problematic ends of our chromosome, which has been claimed to be responsible for the getting old phenomena. But anyway, so that's the thing. And even in these days, also not only this, the violation of the conversion from RNA to DNA, but also protein to RNA. This up until now, this has been something kind of real, more rigorous doctrine. So you cannot violate it. So you cannot make RNA out of protein, but that is also became a thing of past. It is possible to generate RNA starting from protein in these days. So nothing remains unchanged in this field. So getting back to the basic, the point over here, like the way how this transfer of information in this direction is like how to convert this any information which is only the conceptual thing into the real protein building structure is there has to be some very specific relationship or link exists between the genetic the nucleotide, whether it's in the form of DNA or the copied version of such that which is RNA, with the protein. Protein is nothing but the whole combination assembled, which is assembled by individual amino acids. Right. So what's the link? What's the link between the amino acid and the nucleotide? There has to be some kind of a specific combination of nucleotide to designate each particular identity of amino acids. That is the concept of codon. Codon is the set of 3 nucleotides on messenger RNA, and the fact that these 3 nucleotides units present on messenger RNA, but the messenger RNA is actually the copy of the DNA of the coding region of the DNA gene. So we can also talk about the presence of the codon on the DNA as well. But the important thing is whether it's on DNA or messenger RNA, but the codon is a unit that defines an individual single amino acid. So 3 nucleotide NT will specify one amino acid is their relationship and its a significance. So actually historically speaking, when the before Watson and Crick discovered the and then shows the correct structure of the DNA double helix, the majority of the scientific community scientists most of these scientists were in favor of that notion of protein as the genetic material, because the thing that obscured this whether DNA or proteins is the genetic material was because the discovery of the chromosome in chemical composition. Then it turned out that half of this chromosome contain proteins and another half contain the nucleic acids. And chemically, scientists knew that DNA is made of 4 different types of nucleotide, whereas protein is made of 20 different kinds of amino acids. Then naturally, the way scientists solved also protein should be the genetic material, because with the combination of different combination of 20 different amino acids, you can generate more number of diversity, which is the key for becoming the genetic material. Diversity is one of those such the unique characteristic about this the traits. But on the other hand, thinking of this nucleic acid made only of 4 different kinds of nucleotide, then you can think with these 4 nucleotides, how much the variation can you generate out of only 4 different nucleotides? And that was the main reason why the scientists in this field believed for a long time that the protein is the genetic material. But anyway, but the way with only these 4 nucleotides that still DNA can generate so much diversity is this relationship of so this, according to this central dogma, these 4 different types of nucleotides should be able to cover at least 20 different types of amino acids, and then only then these so-called central dogma relationship can exist. So the single codon consists of 3 nucleotides. If that is the case, actually, if you have this type of rule, then the maximum number of differences that can be generated out of this combination thing is actually, it becomes 64 different amino acids. You have only 20 different amino acids, so it's more than enough to cover this. And so, like on RNA, for example, this 3 particular combination of nucleotide AUGU is in DNA, it's replaced with the T, and then it calls for one specific amino acid called the methionine. By convention, name of amino acid is used to specify a certain amino acid. That's 3 letters. Its full name is methionine, for example. Then usually it is used in abbreviation of 3 letters, and 3 consecutive G nucleotide in RNA nucleotide, then it will code for glycine and proline and threonine, etc. So all those 20 different amino acids has its own unique 3-letter designation convention. And actually, in a little bit more advanced scientific community, even this 3-letter is inconvenient. So usually they prefer having one single letter convention. And then if you are a major life science major, probably you better get accustomed to using the single letter the convention of amino acid representation rather than 3 letters. But at this level it's not necessary. So here if you overview this relationship, so if this is the DNA double helix, one of these 2 strands will serve as template DNA, which is going to be a complementary sequence of the messenger RNA sequences. Right? So this messenger RNA here will be an exact copy in terms of this is through the complementary base pairing. So T will pair with A of RNA nucleotide, and G will pair with C, and A will pair with U, like this. Okay. So always the relationship between the template DNA. I just made a mistake. This is not. This is a DNA double helix, double strand of the gene region. So this is called a template DNA, which serves as the complementary, the copy the sequence. Each messenger RNA 3 by 3 specifically designate any one single kind of amino acid, so that ACU will specify threonine and proline and blah blah blah, like this. Okay. So that's settled. Now on DNA, actually the relationship between this template DNA and the other complementary DNA strand is also complementary. So if you take a look at the sequence of the other strand, it goes like ACT, which is identical to the corresponding part of the messenger RNA, except for this T and U conversion. And the CCT sequence, which is going to be identical to that messenger RNA, corresponding part of the messenger RNA sequence, except for this TU conversion, and GAG should be identical, like that. So even without looking at the messenger RNA sequence, you can have a clear idea about what kind of a code, what kind of codons are present in a specific area of the gene just by looking at the other strand of the DNA, which is complementary to the template DNA strand. That's why simply by looking at and simply by reading through DNA sequence, you can figure out more directly figure out what kind of amino acids will be made out of a specific region of the DNA. I mean, DNA is the reason, or because of this reason. And this relationship of a complementary base pair goes further in that actually there is another such complementary base pairing relationship between the messenger RNA and tRNA. What's tRNA? Transfer RNase is the one that actually delivers the specific amino acids to the site of the protein synthesis, which is a ribosome, right? So we will get to that part in a minute, but let's take care of this codon thing first. So here comes that there are 20 different amino acids are used in protein assembly, but in theoretical basis, if you have 4 different nucleotides, then out of these 4, you are going to have 3 to pick 3 out of these 4, and you have 3 order of such events. So number 1, 2, 3. So out of 4, there is 4 possibilities times the second position of this codon, another 4 possibilities, because repetition is allowed, and 4, so it will comes 4 to the power of a third, which becomes 64 different possible, theoretically, combinations. So in theory, that means you can create 64 different amino acids. I shouldn't say create, but you can designate using this rule. You can designate 64 different amino acids is the thing. However, since there are only 20 amino acids that participate in the protein assembly, it is not that we have only 20 different kind of amino acids in the world. There are way more than 20 different amino acids, because well, this I have mentioned earlier, this in the chapter 2 or 3 earlier, when we were talking about the macromolecules. But however, for some reason, only these 20 amino acids are the ones that become a part of this protein built in the life. So naturally, out of this, some of them, because you will generate 64 different kind of codons because of this nature of DNA. The 4 nucleotides will exist in random order on DNA. So if a certain segment of the area of the DNA is going to be used as like a gene, the messenger RNA coding gene, then obviously you will have 64 different possible codons that can be found, usually, in theory. So in other words, these codons should be redundant. In other words, more than one different combination of the 3 nucleotides codon will specify the same kind of amino acid. This is what we call it codon redundancy. So if that was the case, then why do we have this type of waste by having 3 nucleotide codon things? If we, in the first place, if we had like only two nucleotides to specify on a single amino acid, like two nucleotide codon system, it would have been way like a less waste of in these issues. No, it is not. So if we decide to make these two nucleotides to have one single codon unit, then in this way you have this, and only 4x4, there is only 16 different possibilities, so this is not enough. So that is the reason why the 3 nucleotide codon is the minimum ever possible to cover all 20 different amino acids using this scheme. On the other hand, if you somehow decided to, as an organism, somehow decided to use a 4 nucleotide as a one codon unit, then it is like the possible combination become 256 different ways. So that is the whole too much of excessive waste. So that is what it is saying. So now you have this 3 nucleotide as a codon. How actually in practice each codon have this kind of matching specific relationship with a specific amino acid? There comes the action of the transfer RNA. So in each different transfer RNA, there is a specific area which is complementary to that to a given combination of a codon. So that's why its name is anticodon. So if this was the messenger RNA codon, the reason why this ACU can match to this amino acid threonine is because this tRNA, this is a tRNA, a rough structure of a tRNA, right? And each tRNA has specific amino acid, and in this case, this particular amino acid happens to be this threonine, and this tRNA at specific location, usually the bottom of this tRNA structural loop, tRNA looks like this. Okay. Oh, it's ugly. Why suddenly I am just such a poor drawer. So UGA, this particular anticodon sequence, which is complementary to this particular codon, A is complementary to U, C, G, and UA, just like that. And that's the reason why the specific relationship between this codon and amino acid threonine can exist. Right? So once again, in the case of CCU of this particular codon have a specific designation of a proline amino acid is because this particular transfer RNA shaped like this and these are complementary base pair, and at the end of one, this is a 5' end of RNA and this is a 3' end of RNA, and here comes another amino acid, and that amino acid happens to be a proline. In other words, there has to be at least 20 different transfer RNA, each of which has specific carrying amino acids. But since the codon and anticodon relationship exists, the number of actually different transfer RNA, by virtue of having these different anticodon sequences, should have, at least theoretically, should be 64. In that way, each of 64 different possible codon would have exact matching anticodon on each different type of transfer RNA. However, the group of, in terms of speaking of the group of different transfer RNA, there should be only 20 different kinds of transfer RNA in terms of what type of amino acids are being carried by a given type of transfer RNA. That is the thing. Okay, so so this is more realistic model of transfer RNA. This is a three-dimensional model structure of the transfer RNA. And at the end, this 3' end, there is an amino acid will be hooked to the end of this area. And at the bottom of this loop is the area you can find the anticodon. The 3 nucleotide anticodon sequence. So if this transfer RNA is the one carry the tryptophan amino acids, then the anticodon sequence happens to be ACC, which would be complementary to the codon sequence of messenger RNA, which is what? U, G, and G is. But remember, if this UGG sequence is in order of 5' to 3' end, then this relationship should also be anti-parallel, so that this transfer RNA sequence direction is 5' and this is a 3' end. So whenever this complementary base pairing create this interaction of a two-strand, even if it is a local two-strand, double-stranded structure between DNA, two DNA, or one DNA and RNA, or between two RNA like this, the orientation should always be anti-parallel like this. So each transfer RNA carries a single anticodon, and the type of amino acid is carried by this specific transfer RNA is actually determined by such characters. But here, interestingly, instead of 64, there are only 61 different transfer RNA in terms of this the anticodon. Different anticodon sequences. So here there are 3 codons that do not have any matching anticodon carrying transfer RNAs. So is this some kind of an aberration? No, it turns out that it is actually a blessing. So these 20 different amino acids will be like out of this 61 different transfer RNA, some of them, multiples of different tRNA will carry one specific type of transfer, the amino acids, in that it has this type of, so those relationships can be organized, summarized in this codon table. So for example, in the case of a proline, 4 different types of codons, which means there are 4 different types of transfer RNAs carrying each of these anticodon sequences, all of which happen to carry the same amino acid proline. So there are 4 transfer RNA, 4 different types of transfer RNA will pick up same proline, but each of these 4 different transfer RNA have a different type of anticodon sequences, each of which matches to this respectively CCU, CCC, CCA, CCG codon sequences on the messenger RNA. So that's how this 4 nucleotide codon can specify a single proline amino acid. And in the case of the valine amino acids, the same thing goes. 4 different codon each of which have a matching anticodon sequence of the transfer RNA, all of which will carry the same valine amino acid, just like that. And so these are the example of such a codon redundancy. But since there are 61 anticodon versus 64 codon, unfortunately, these three, UAA and UAG and then UGA, these three codons do not have the corresponding anticodon sequences. None of these transfer RNA exist having this particular anticodon sequences. And then thanks to that, either one of these three combinations of codon, UAA, UAG, UGA, obviously none of us need to memorize these three codons that are used instead of specifying the amino acid, but they are rather used as a signal, stop sign, which means when this whole protein building procedure goes along the messenger RNA, when this ribosome structure is encountered with finally encountered with one of the three, either one of these three codes, like either UAA, UAG, or UGA, even though you don't have to memorize, but like soon enough you will get to memorize if you just are in this business with UA, UGA, UA, it's relatively not difficult to stay in your brain. Then, since there is no matching transfer RNA, so there's no further movement can be processed at this point, and then virtually this protein building procedure will stop there, and that's why these three nucleotides act as a stop signal, so they are called the stop codon. Okay? And so the actual thing, although we don't really have to worry too much details about the actual process, which is actually complicated things. And so this amino acid is with the spending of an energy, this amino acid will be attached to one end of this transfer RNA, and only the transfer RNA having this attached to the amino acid can enter the site of a protein synthesis in the ribosome. Okay. So for the amino acid, there is two rooms on the ribosome. So in other words, two transfer RNA carrying, each of which carrying specific amino acid, can enter and then stay at the same time, stay in the specific location of the ribosome is the key. So when both amino acids carrying transfer RNAs are present, and then the two amino acids can be combined through the chemical bond formation, which is the peptide bond. One of those condensation reactions. And the reason why each specific amino acid can stay on and attach to this specific location is the relationship between codon and anticodon complementarity relationship. Without it, any given transfer RNA cannot be attached to a specific location of this messenger RNA on the ribosome. So in this way, the ribosome, this is a starting part of the messenger RNA, which is a 5', and then let's say this is the end, 3' end of the messenger RNA, and ribosome will walk through this messenger RNA in working at each step, it takes 3 nucleotide and 3 nucleotide. One step will make this ribosome advance to 3 further codons, nucleotide on the messenger RNA. So by doing so, when in a given like snapshot, if you take a snapshot, in a given moment when these 2 amino acids are present, and one amino acid, the pre-existing amino acid, already have several amino acid attached to it due to this repeated procedure of transferring one amino acid to the other. And now the newly combed, this is a new transfer RNA carrying also another amino acid. Then the direction of the transfer of this amino acid through peptide bond is always from old one to new one. So whole entire amino acid will be transferred into this new amino acid. And that makes sense because the ribosome will move. Next step, the ribosome will move the 3' downstream by 3 further nucleotide of another codon. That means within this remaining available 2 rooms, this existing, the old amino acid will be kicked out. So if the amino acid, existing growing chain of amino acids was still attached to the old one, then you lose everything by then. So this is such a smart move in doing so, of transferring the existing old long chain of growing amino acid will be transferred into the newly combed amino acid on the new transfer RNA just came in. Okay, and it will be repeated. So until when? Until this ribosome arrives at a specific codon, so this stop codon is better be present at the end of these genes. Somehow by accident, if this stop codon is somehow inserted in the middle of the messenger RNA, then you are in trouble because the kind of protein that is made is only halfway through. It's incomplete protein, right? So then, only then, this procedure. By the way, this whole protein generating procedure is what is it called? Translation. This translation process now then stop. Why? Because here ribosome waits for a moment until the new transfer RNA, which carries the anticodon sequence to this stop codon, but as we all know by now that there is none. So this will remain empty for a moment. And this whole ribosome is not such a very patient guy or lady. So it cannot wait any longer, and then this is assembled, its whole structure, and so that's the end of the process of translation. That's how this stop codon function. So no tRNA having anticodon sequences. So there. So these are the one serving as the stop codon. So that's a brief, but all the necessary information with which we can explain the core nuts and bolts of the translation process. The next, we can think about what happens if the whole procedure goes wrong. So what if I have some sequence alteration in our messenger RNA sequence? Actually, if you have this sequence variation as a result of a mutation on the DNA coding region, then it will affect all those copies, corresponding copies of messenger RNA, and then it will affect the products, the protein products. So that's the result, consequence of the mutations, and we are going to take a look at here, brief the principle, nature of the mutation and different kinds of mutation in terms of this, the DNA sequence alteration and how it will affect in the coding, the protein coding event is the key. And then finally take a look at the three different real life example of such mutation is the organization of this part. But obviously I have already spent too much time, so it's very unfortunate, but I have to stop here and have to wait the completion of this discussion until next week's lecture, which is our final lecture of the course. So it's a shame, but yeah, I'm not going to force anything. So I will stop over here and have a nice remaining weekend. Bye bye.